Hello again. The nervous system is one of the major control organ systems of the body. It is able to regulate, control, and command all parts of the body because of its highly specialized cells, the neuron, that is a highly communicative cell. How it is able to do so is the main goal of the series of video lectures you are about to hear in the e-learn. But first, we cover the fundamentals about the nervous system and its cells. And then we talk about the special property of charge separation across the membranes of neurons that enable it to generate signals, which we call here as membrane potential. At the end of this short video lecture, which is the part 1 of the nervous system, you're expected to describe the major divisions of the human nervous system, describe the basic structures of the neuron and its structural and functional types, describe the other cell types of the nervous system, and describe how the resting membrane potential of the neuron is established. The various parts of the nervous system are intimately interconnected, but for convenience, we divide them into two parts. The central nervous system, which composes of the brain and the spinal cord, and the peripheral nervous system, which consists of the nerves and ganglia, as well as its connected sensory organs, special sensory organs, and nerve endings throughout the body. The functional unit of the nervous system is the neuron. It is a highly specialized cell for communication or for signaling, and its structure is designed in such a manner to achieve the power of communication. The main body or the cell body of the neuron is usually stellate, and the surfaces are thrown off into cytoplasmic extensions, many of which are short and branching, which we call as dendrites. This region of the cell body of neurons is the signal receiving region of the neuron. This is where most of signals from other neurons and from other cells, including intercellular messengers, are received by the neuron. Somewhere at the center of the cell body is the nucleus and all its other cell organelles. It has a highly specialized and endoplasmic reticulum that synthesizes many of the protein chemical signals that the neurons release to other neurons or other cells nearby, which are called neurotransmitters. There is one cytoplasmic extension, usually of neurons, that is very long, and we call this the axon. This is the signal transmitting region or part of the neuron. The initial segment of the proximal end of the axon usually is conical, which is also called axon hillock, and usually this is where most of the signals in the form of action potentials are generated. There is a high abundance of sodium chemically gated and voltage gated sodium ion channels and potassium ion channels that are concentrated in this initial segment. Hence, it is able to generate action potentials or signals in this segment of the neuron. The longer half or the longer portion of the axon is cylindrical, but towards the ends of this axon are arborizations or branchings that which we call generally as the axon terminal. When these arborizations nears the next cell or neuron, these axon terminals form bulbous structures, which we call as terminal knob. This is a specialized region that makes contact with other neurons or other cells where they transmit their signals in a very specialized mechanism. This neuron that is shown here is a multipolar neuron. It's one of the structural classification of neurons. It has several branches or several cytoplasmic extensions, since it's called multipolar. There are also neurons that have only two cytoplasmic extension, one being the dendrite and the other being the axon. We call these neurons as bipolar neurons, having only two cytoplasmic extensions or fibers. These bipolar neurons are much more numerous in specialized sensory tissues of the nervous system, especially at uh, retina and that of the olfactory tissue of the nose. Um, there's also a particular kind of structural classification of neurons which has only one fiber or one axon, and we call this as unipolar neuron. But as soon as the axon projects from the cell body, this axon divides into two branches. No? One of these branch extends towards the periphery of the body, and the other branch shortly terminates in the central nervous system. This is what we call unipolar neuron, and this is very classic of many sensory or afferent neurons. Another group of cells that may be found in the nervous system are the glial cells or neuroglia. Sometimes because they generally establish the environment of the neuron that makes the neuron able to function well, the glial cells are also called supporting cells. They are the most diverse group of cells in the nervous system. Many of them are found in the central nervous system. For example, you have astrocytes. The astrocytes are highly stellate cells that hold on to neurons and surrounding glial cells in the nervous tissue. They form a scaffold that supports all the other cells in the nervous tissue. You also have ependymal cells that are highly phagocytic, and they act like the immune cells of the nervous tissue. They clean up the environment of the neurons from unwanted debris. Then you have also oligodendrocytes. These are myelin-forming cells that form coverings of the nerve fibers of neurons in the central nervous system, the oligodendrocytes. And then you have the ependymal cells. The ependymal cells form a lining epithelium at the surfaces of the ventricles or cavities of the brain and spinal cord. They produce cerebrospinal fluid that bathes the entire nervous tissue of the center of the nervous system. At the peripheral nervous system, you also have satellite cells that help cover the specialized cell body of sensory or afferent neurons. And then you have the Schwann cells. The Schwann cells are glial cells that provide myelin sheath covering over the axons and dendrites of axons of sensory or afferent neurons in the peripheral nervous system. They are the counterpart of oligodendrocytes in the central nervous system. The axons of neurons are also called fibers, and most fibers of neurons are covered by myelin sheath, which are actually the membranes of oligodendrocytes in the central nervous system and the Schwann cells in the peripheral nervous system. Because of this myelin sheath, which is formed by the either oligodendrocyte or Schwann cell, the entire fiber is almost completely insulated except at certain regions in between the glial cells. These barren regions or these uninsulated regions are called nodes of Ranvier. These are very important regions along the fiber or axon of many sensory and motor neurons, because this is where most of the signals jump from one node of Ranvier to another in a saltatory fashion. This slide shows or diagrams the functional classes of neurons. That includes afferent neurons, efferent neurons, and the interneurons. The afferent neurons transmit signals from the periphery of the body or from specialized sensory receptors towards the central nervous system. Their cell body and long processes of axon lies in the peripheral nervous system. Only a short part of their axon lies in the central nervous system. And at the central nervous system, they terminate and make special contact with cell bodies of interneurons in the central nervous system we call synapse. The afferent neurons are also known as sensory neurons because many of the sensory organs have afferent neurons that gather special informations that they transmit to the central nervous system. The efferent neurons have their cell bodies and a short part of their axons in the central nervous system, but the longer part of their axons lies in the peripheral nervous system, where they terminate in specialized contact points with muscles and glands and other neurons, which we also call as synapse. Their endings of their axons are also known as axon terminals. The efferent neurons are also called motor neurons because once they have signals coming from the interneurons of the central nervous system, these signals are actually command signals to effector organs and tissues such as muscles and glands. So they generate action or responses in the effector cells, hence they are called motor neurons. And then finally, you have the interneurons in the central nervous system. They lie mostly in the central nervous system. The interneurons make up 99% of all neurons in the nervous system. And they form circuits, specialized circuits, that integrate all the sensory informations coming from the afferent neurons and then generate signals and commands that they transmit back to the efferent neurons. Many of reflexes are formed by an array of afferent neurons and interneurons and efferent neurons. The slide here shows the general functional organization of the nervous system, wherein you have informations gathered by sensory receptors or special sensory organs that are then sent as sensory input to the central nervous system, where these sensory informations are gathered and integrated in many circuits where a particular and appropriate command for response is generated. And these commands are then transmitted to various effectors of the body that includes muscles and glands and other specialized neurons in the peripheral nervous system via the motor output. For us to understand the resting membrane potential that stores energy for signaling by the neuron, we need to understand some basic principles of electrophysiology, wherein charges of the same type repel each other and opposite charges attract each other. The charged particles will tend to move toward each other if they are opposite charges, and that is if they are not separated by a barrier. In the natural setting of the nervous system, however, there is a natural barrier that separates the ionic or charges in the nervous system, and this is made or formed by the plasma membrane of neurons. The plasma membrane of neurons is selectively permeable to certain ions that generates a separation of charge across the membrane. which is known as the resting membrane potential. This potential is called electrical potential because it is determined by the difference of charges across the membrane. The units of electrical potential is volts, but because the total charge that can be detected in biological system is so small, these potential differences are measured in millivolts. 1 millivolt is equal to 0.001 volt. At rest, neurons have a potential difference across their plasma membranes. When we place electrodes at the outside and then another electrode at the inside, we will find with a voltmeter that there is a charge separation across the membrane. And this charge separation is usually measured to be about negative 70 millivolts at resting membrane potential. The resting membrane potential of neurons is ascribed or attributed mainly to two factors. That is the difference in specific ion concentration across the membrane, as shown here by this table, and the difference in membrane permeability to the different ions, due in turn to the presence of different ion channels. Usually, the membrane of neurons have more open ion channels for potassium than for sodium ions. Now, this table here shows the distribution of major ions that are distributed across the plasma membrane of a typical nerve cell. So, as you can see here in this table, there are more sodium ions and chlorine ions that are concentrated at the extracellular side of the membrane than they are at the intracellular side, measured in millimoles per liter. Whereas for potassium, they are more concentrated inside or intracellularly than outside the plasma membrane of neurons. And because of this difference of concentration, and they have a tendency to diffuse across the membrane, they generate a potential difference, which we call as resting membrane potential. But let me emphasize here that this charge difference across the neuron is only localized at the surfaces, the inner or cytoplasmic side of the membrane and the outer or extracellular side of the neuron's membrane. Elsewhere, inside the cytoplasm of the neuron and outside the extracellular fluid of the neuron, the charge is neutral. But this membrane charge separation is localized only across the immediate vicinities of the plasma membrane. This slide here shows the theoretical establishment of a membrane potential that is affected by concentration gradient of ions across a selectively permeable membrane. This series of images here of ionic distributions across a selectively permeable membrane describes how the membrane potential is established, that is in turn produced by concentration gradient and electrical potential or membrane potential. Now, let's take this example here at this particular side, wherein you have two compartments with two different solutions but of equal molarities. You have sodium chloride and potassium chloride in compartment 1 and compartment 2 respectively. The membrane or the barrier that separates the two equally concentrated solutions are permeable mainly to potassium but not to sodium. We will discount the uh, concentration of chlorine here because they are already of equal concentrations. So their movements will not affect much of these conditions. But because the barrier across these compartments 1 and 2 is more permeable to potassium, and potassium is concentrated at compartment 2 than at compartment 1, potassium will tend to diffuse into compartment 1 because of concentration gradient. Sodium also has a tendency to diffuse in from compartment 1 to compartment 2, but it cannot do so because there is no permeability of the sodium, or rather of the barrier for sodium. And so sodium stays within compartment 1. But as potassium diffuses along its concentration gradient from compartment 2 to compartment 1, it contributes to the positive charge across the compartments, making compartment 1 more positively charged than compartment 2 as more potassium leaves the compartment 2. So there is a charge difference now that is established, initiated first by the concentration gradient. But as more and more potassium moves across the membrane from compartment 2 to compartment 1, these potassium ions that are diffusing eventually slows down because of the charge that is building up at compartment 1. Because potassium is positively charged and the interior side of compartment 1 across the barrier is becoming more positive, a few potassium can move in and more potassium will begin to move out of compartment 1. And as the magnitude of diffusion of potassium into compartment 1 is balanced by the diffusion of potassium back to compartment 2 because of repulsion of charge, membrane potential is established wherein the compartment 1 side of the barrier or of the membrane is positive, whereas the other side, the compartment 2 side of the barrier or membrane, is negatively charged because of the greater potassium diffusion from compartment 2 to compartment 1. And so you have a membrane potential that is established, wherein the magnitude of diffusion of potassium from compartment 2 to compartment 1 is balanced by the magnitude of diffusion of potassium from compartment 1 back to compartment 2. This is a condition which we call as equilibrium potential. The same thing could happen if we have a solution, or rather two solutions that are separated by a selectively permeable membrane that is now only permeable to sodium than to potassium. Sodium will tend to diffuse from compartment 1 to compartment 2 because of concentration gradient. But as more sodium diffuses from compartment 1 to compartment 2, this creates a charge separation. The inner side of compartment 2 membrane becomes more positive because of the influx of sodium, and the side of compartment 1 membrane, it becomes negatively charged because more sodium ions are leaving compartment 1. Now, there comes a point when the diffusion of sodium from compartment 1 to compartment 2 and from the diffusion of sodium from compartment 2 to compartment 1 will be equal or zero. At this point, equilibrium potential between sodium at compartment 1 and compartment 2 is established. But as you can see, At this equilibrium potential, there is charge difference or membrane potential that is established because of the different concentration gradient and the different permeabilities of the membrane to different ions. For a given concentration gradient, the greater the membrane permeability to one type of ion is, the greater the contribution of that ion will make to the membrane potential. Um, Forces acting on sodium when membrane is at resting membrane potential and sodium equilibrium potential is shown here. When the forces acting also on potassium when the membrane is at resting potential and at the potassium equilibrium potential is shown here as well. At resting membrane potential of negative 70 millivolts, both the concentration and electrical gradients favor the inward movement of sodium, whereas the potassium concentration and electrical gradients are in opposite direction. The greater permeability of potassium in opposite directions maintains the resting membrane potential at a value near equilibrium potential. So this therefore means that the resting membrane potential is more dependent on the movements of potassium. The steady state of sodium and potassium movements due to passive diffusions is also aided by the active transport of the same ions across the membrane. There is an active transport called sodium-potassium pump that is abundant at the membrane of neurons. Okay, so this sodium-potassium pump usually pumps 3 sodium ions out of the neuron and into the extracellular fluid as it pumps 2 potassium ions back from the extracellular fluid back to the intracellular fluid. This helps maintain the separation of ions across the membrane. But other than that, you have also the relative permeabilities of the ions, or rather of the membranes of neurons to the sodium and potassium ions. The membrane of neurons have more ion channels that are passive and open for potassium than there are for sodium. So there are more potassium ions therefore that diffuses out of the membrane than there are sodium ions diffusing into the membrane. Hence, because of these larger potassium ions diffusing out, you have a slightly negative charge across the inner side or the cytoplasmic side of the membrane than the outer side, which is positively charged. Hence, you have a membrane potential and separation of charge that is established both by the differential concentration gradient and the relative permeability of the membrane, plus the action of the sodium-potassium pump that is an active transport. The next topic in the series will be about membrane potential changes that are used by the neuron for neural signaling and the mechanism of neural synaptic transmission. Stay tuned. Thank you.